like ear, eye, tongue, all the spatial sensation organ. Uh, and in this, uh, after that, we have third midterm exam. So today we don't have a lot. We will just talk about the histology of the nervous system. Okay. So let me go to the canvas and find out the study area for the histology of nervous system.
So the structures we would just be studying for this chapter is only in histology.
Yeah, today only in the histology, because the first chapter is the nervous tissue and the overview of the nervous tissue.
Okay.
So, and I have already posted the homework for that chapter. So as usual, you go to your canvas, and then in canvas, you go to study area. And in the study area, in the canvas, My Lab Mastery, open My Lab Mastery. And then study area, launch the study area.
Oh, by the way, we can't see the screen if you're trying to
Yeah.
show us.
No, I have done that. I'm going to
Oh,
say it at the end
okay.
because I'm doing that thing every day. So Paul, and then histology, and then nervous tissue. Okay. So if you go back on the home of Paul, let me share this stage. Okay. So hopefully you all are there. This is the histology section. You guys can open on your own computer too, or you can share with me. So here in the histology section, we have all the systems, and we're going to go nervous tissue, and then we'll go over each of them. So first of all, nervous system contains the organ, which is the brain, spinal cord, then nerves, ganglia, and receptors. Those are the part of nervous system. Nervous system is divided into two groups. Let me write down here and notate. Uh, so nervous system has the organs are the brain, the brain, spinal cord, nerves. We have two kinds of nerve, spinal nerves and cranial nerves, ganglia, and receptors. These are the part of nervous system. Okay. Nervous system organs contains two kinds of cells, and they are the neuron and supporting cells. Supporting cells are like 5 to 7 times more common than the neuron. Okay. So this is the nervous system organs, whether it is brain or a spinal cord or ganglia or anything like neuron. They all contain two kinds of cells. They have neurons and the supporting cell. So we're going to see how they look like in these organs. That is all the histology of the nervous system is. Okay. So here we have 24 slides. And then we're going to go one by one. So here is some of the terminology in the nervous system. What is neuron? What is nerve fiber? What is nerve? So neuron is nerve cell body, neuron cell. And neuron cell is like any other cell which has plasma membrane, cytoplasm, and organelles. Whereas the nerves are the bundle of axons of the neuron. Okay, that is called nerve. Okay, and similar bundle of axons in the central nervous system, like in brain or a spinal cord, we don't call them nerve, we call them tract. That is called the tract. Okay, so let's see here the structure. So new nervous systems, neurons can be classified based on their structure or their function. Okay, the function wise, a neuron can be sensory neuron or motor neuron or interneuron. So sensory means which carry impulse to the central nervous system. Motor means the neuron which brings impulse, electrical impulse from the central nervous system to the periphery. And interneurons are neurons which is between them. Similarly, based on their structure, neurons can be classified into the unipolar, pseudo-unipolar, multipolar, and bipolar neuron. That means how many poles they have. If there is like one axon, one dendrites, one cell body, then that is bipolar. If there is one axon, several dendrites and cell body, then that is multipolar. And pseudo-unipolar means they have one cell body, but one axon then divide and becomes two. So these are all in the lecture. I'm not going to go over here. I'm just summarizing them. You can see them in the lecture. Okay. So I'm going to talk here the multipolar neuron. If you see this slide, okay, so let's see here in this slide, you see, I'm going to erase this. Okay, so in this histology slide, if you see here, you see very minimum number of neuron cell bodies, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. But you see the small dots? Those small dots are the nuclei of supporting cells. So you can imagine here, you can see here how many more times there are the neuroglia or the supporting cells. Another name for supporting cells are neuroglia in central nervous system. Okay, so the small dots are the nuclei of supporting cells. These are the neurons. In the neuron, you see this is one long extension of the plasma membrane that is called, what is that called?
Axon.
Axon. And then the small ones are called dendrites. Okay. Highly magnified here, you can see everything very easy. So you see dendrites and then axon here. The large one are axon, all the smalls are dendrites. And inside you see the cell body. The cell body is also called soma. Inside the cell body, you have all the structure which are like other neurons. You have neuroplasm, which is called the cytoplasm, then organelles, then nuclei, nucleolus, everything. Okay, all the structure of atypical cells. And again, you can see all supporting cells and nuclei. Same thing here. See, the neuroglial cells, dendrites, nucleus, nucleolus, cell body, and then axons and axon hillock. This axon hillock is functionally important because this is the area where axon potential generation starts. So when somebody is stimulated, like the dendrites bring the impulse towards the cell body and axons takes away from it. So when you stimulate the dendrites, then all the depolarization, the change in electrochemical status of the plasma membrane of the ions, they gather here. And in the plasma membrane here, there is a lot of sodium channel. So a lot of sodium channel open, sodium get inside, and then axon potential start from here. That's why axon hillock is important. Axon hillock is important point in a neuron to generate axon potential. Okay. There is multipolar neuron with better staining, silver staining here. And you see the same structure. See the axon, neuroglia, dendrites, axon hillock. And now in this cells, so I said nerve fiber, teased preparation. Nerve fibers, what is nerve fibers? Nerve fibers are bundle of axons, and these all axons are separated, and then the GM size staining, or I think silver is staining. This is silver is staining, and then you can see here. So you see nerve fiber, and they have myelination outside, and there are some gap in the nerve fibers. You see here from here, and you see there is gap. Here is gap. Those gap is called nodes of Ranvier. In this gap region, there is no myelination. They are unmyelinated region of the axons. This is the nerve section, longitudinal section. So in our body, you can take some example of the nerve, like you have axillary nerve in your axilla. You have sciatic nerve on your back, from your upper back goes into your thigh, which is a bundle of action, which is connecting your thigh muscle, leg area to your brain, to your spinal cord, okay? So if you take those nerve and make a longitudinal section and then stain, you're gonna see this structure. Because nerve fibers are axon and they are very tender. So to protect them, they have like several layers of covering like muscles. If you remember muscle, the individual muscle cell is called
Muscle fiber.
muscle fiber, and muscle fiber is covered by connective tissue, and that is called
Perimysium.
nope,
Endomysium.
endomysium. Okay. Similarly, each individual axon has myelination, and after the myelination, there is a layer of connective tissue which covers, which is called endoneurium. And then more than one muscle cell make a bundle. We call them fascicle. And fascicles are covered by
Perimysium.
perimysium. And similarly, nerve fiber fascicles here is also covered by perineurium. Then whole nerve, for example, sciatic nerve. is also covered by another connective tissue, and that is called, if this is perineurium, then that will be epineurium. Okay, so exactly like muscle fiber, nerve fibers are also covered by several layers of connective tissue to protect them. That's why when you are running, exercising, your nerve fibers are not ruptured easily, not broken down, not torn away easily, because connective tissue are protecting them. Okay, so you see here fascicles, several fascicle, and then outside the fascicle there is the perineurium. Again, fascicle, perineurium, and then inside there, in the fascicle, there is like several axon. One of the axon is here. So every axon is under the endoneurium. There is a fatty sheath covering, and that sheath is produced by a supporting cells in the peripheral nervous system. That is called Swan cells. So you see Swan cell nuclei. These Swan cells are making the fat around the axon, and that is called myelin. Myelin. And then in the myelin, you have some of the nodes of Ranvier. See, nodes of Ranvier, where there is gap, there is no fatty sheath covering. So if I take a nerve, let's see if you have the sciatic nerve, and then you cut the sciatic nerve in a transverse section and then see under microscope. So as I said earlier, you can see everything. So inside you have the nerve fiber, and bundle of nerve fibers are called fascicle. Around the fascicle, there is connective tissue that is called perineurium. And then several fascicles are covered by other connective tissue that is called epineurium. Got it? So inside, if you go inside in this fascicle, you see the white, the area covering, those are endoneurium. Here you go. Endoneurium. Endoneurium covers every individual nerve fiber. And under the endoneurium, you have myelin sheath. And under the myelin sheath, what do you have? What do you find under the myelin sheath?
I think it's the nerve fibers because it's kind of myelinated.
Yes, it is nerve fibers. It is axons.
Mm-hmm.
Yep. Okay, same thing here. This is a longitudinal section, not cross section, longitudinal section. Swan cell, myelinated nerve fibers, nodes of Ranvier. Now, let's see. So up to now, we have seen all the nerve fibers. Let's see the brain part and then see how they look. So brain is also made up of neurons and the supporting cells. Here is cerebellum. Cerebellum is the part of your brain which is in the posterior part, and this structure is responsible for balancing of your body, which is called equilibrium. Okay, so this is cerebellum, silver stained sagittal section. In the brain, if you make a brain cut section, particularly your cerebrum, then out part is gray matter and inner is white matter. But here in the cerebellum, that's just opposite. You see, the inside is gray matter. Gray matter looks gray because they are mostly made up of neuron cell bodies. And outer part is white matter because they are made up of nerve fibers, which means they are made up of axons of the neuron. Okay, and in the brain there is like the enfoldings, and there is bump and enfolding. So enfolding is called sulcus, and bumps of the brain surface is called gyrus. Okay, so If we magnify, you can see the gray matter, the sulcus, and the white matter. White matter is outside in the cerebellum. But if you go to cerebrum, which is the upper part of the brain, it will be just opposite. Spinal cord will be also like cerebellum. Okay, so gray matter and the white matter. Gray matter is, you see small dots, those are the cells. These cells are called granular cells. These look like granules. Okay. Now, a spinal cord in the cervical region. When you cut a spinal cord, as I said earlier, the histology of a spinal cord is like cerebellum, not like cerebrum. So outside part, these all are white matter. That means all the axons bundles is coming from your brain and going downwards, or from outside it is going up like a highway. And these white matters have several names. So you see, your spinal cord is like your brain. We have two hemispheres. You see, this is the dorsal median sulcus and ventral middle fissure. And then you have exactly same mirror image of brain, the spinal cord. So right half of the spinal cord is exactly like left half of the spinal cord. And in the central, there is a hole that is called central canal, which contains cerebrospinal fluid, CSF. So outside is all white matter, and white matter have different names. We call them funiculus. This is dorsal finiculus. This is the lateral finiculus, and this is ventral finiculus. In the center you have, you see the butterfly like a structure, or you can say it is like letter H, and that is gray matter. This gray matter contains all the cell bodies of the neuron in the spinal cord. Okay, and they are like horns, so we call them, the posterior part of the gray matter is called dorsal horn, anterior is called ventral horn, and there is some which is not showing here, there is lateral horn too. Okay, in different region of the spinal cord. And then outside the spinal cord, you see there is all the connective tissue, meninges, bone, everything. So same structure, highly magnified here. Dorsal funiculus, dorsal horn, lateral funiculus, lateral horn, ventral horn. Okay, lateral horn, they are not. They are like mostly in the lumbar region. So we are magnifying here in the horn region, interior horn, and you can see in interior horn there is central canal, and then gray commissure. Gray commissure is the gray matter which connects right hemisphere of the spinal cord to the left hemisphere. Okay, you see ventral horn, and then you can see the cell bodies inside it, you see, and then supporting cells, blood vessels, and other stuff. Same thing again here, thoracic region. Dorsal funiculus, I think pretty much same thing here too. Let me tell you some function-wise structure here. So dorsal funiculus are the nerve fibers which are sensory nerve fibers. Okay, ventral funiculus are nerve fibers of motor neuron. Okay, lateral funiculus are mixed. Okay, the ventral ventral horn are the cell bodies of motor neuron, particularly somatic motor neuron. Lateral horn, which is not showing in this diagram, are the area where there is cell bodies of autonomic motor neuron. And dorsal horn are the cell bodies of the sensory neuron. Okay, so that's how these all area. So let's see if somebody has spinal cord damage, and if you have damage here in this area, gray commissure, ventral gray commissure, that means your skeletal muscles, you cannot move your limbs because your muscles are paralyzed or very stiff. Okay, same thing here, all the structures, ventral hold, central canal, gray commissure. Now highly magnified here, you see the spinal cord, somatic motor neurons of the ventral horn. So we are just taking ventral horn of the spinal cord and magnifying, and you can see the neuron there, the dendrites and axons. You can see the neuroglial cells. Neuroglial cells are supporting cells. So what are the neuroglial cells? Neuroglial cells are, let me write down. Neuroglial cells are in the CNS. They are astrocytes, oligodendrocytes, ependymal cells, and macroglia. In PNS, there are only two. There are only two supporting cells, and they are Swan cell and satellite cell. So what are their functions? Astrocytes create blood-brain barrier. Oligodendrocyte myelinates axons in the central nervous system. Ependymal cells lines the cavity, ventricles of the brain and filter CSF. Macroglia are phagocytic cells in the central nervous system. In the peripheral nervous system, PNS, Swan cells myelinate the axons like oligodendrocytes in the central nervous system. And satellite cells wrap around the ganglia. Okay? So that is the supporting cells. Here you go. So in this diagram, you can see this is the peripheral nervous system ganglia. Cell bodies of unipolar neuron. So ganglia in the peripheral nervous system, ganglia is cluster or group of cell bodies of sensory neuron. So you see these are the group of cell bodies, and then around it, these are the satellite cells, which is satelliting around the ganglia. Here, it's a better diagram, dorsal root ganglia. You can see these cell bodies of unipolar neuron in the ganglia, and the satellite cells are around it. Okay. So these are all in the histology of the nervous system. So basically we talked about what is neuron, what is nerve fiber, what is tract, what is ganglia, and what is nuclei, okay? And then all the structure of the nerve fibers. Do you guys have any question? I'm gonna stop recording here.